Welcome back friends, welcome again another video tutorial of A to Z biology. Today's discussion is glycolysis. What is glycolysis? Glycolysis, the another name of glycolysis is called Embden-Meyerhof-Parnas pathway or EMP pathway. Glycolysis is a metabolic pathway. Glycolysis is a metabolic pathway. What type of metabolism is it? Metabolic pathway. Glycolysis is a metabolic pathway. Metabolic pathway are basically two types: catabolism and anabolism. Catabolism means breakdown of compounds and anabolism means synthesis of any kind of compound. So in case of glycolysis there is a breakdown of glucose. So it is a catabolic type of metabolic pathway. What is it? It is a catabolic type of metabolic pathway. Why we break glucose? Glucose is the most preferable energy source. And by breaking the glucose we can produce energy in our body. So we break glucose. So this is the importance of glycolysis. The site of glycolysis is glycolysis takes place all cells. Glycolysis takes place all the cells inside the cytosol. Cytosol is the gel-like matrix of the cell's interior, and glycolytic enzymes are present in the cytosol. Glycolysis takes place anaerobically. Anaerobically means oxygen deficient condition, or aerobically in oxygen rich condition or oxygen state condition. In these two conditions glycolysis takes place and gives us energy. All the cells of our body, all the living cells carry this glycolysis process. Another major importance of glycolysis are in case of red blood cell. In case of these red blood cells or RBC, glycolysis is the sole source, or glycolysis is the major source by which RBC produce their energy. Because RBC does not contain mitochondria, so oxidative phosphorylation is not occurred. So ATP is not produced by other sources except glycolysis. So in case of our mature RBC, glycolysis is the major source of energy production. In glycolysis, carbon skeleton of different kinds of amino acids, non-essential amino acids are formed via this glycolysis process. The actual definition of this glycolysis is one molecule of glucose breaks by different kinds of steps and ultimately form two molecules of pyruvate. One molecule of glucose breaks and form two molecules of pyruvate in different steps, and in this step there is a production of ATP. We discuss the entire step of the glycolytic process. And glycolysis, another important thing is that glycolysis is poor in cardiac muscles. Anaerobic glycolysis is poor in cardiac muscle. So in case of ischemia, cardiac muscles are very prone to damage or very prone to necrosis. Cardiac muscle, in the cardiac muscle, glycolysis is very poor. Another thing about the Glycolysis. So different kinds of enzymes participate in this glycolysis process. So the reversible steps of the glycolysis, reversible steps of the glycolysis is involved in gluconeogenesis process. That means formation of reversible steps of the glycolysis involved in gluconeogenesis. Gluconeogenesis is the formation of glucose from non-carbohydrate sources. So actual glycolysis, this is the glycolysis. Glucose fast enters into the cell via different kinds of glucose transporter, and this glucose entry is dependent on the insulin concentration of our blood. This glucose entry is dependent on the blood concentration of insulin. So we first intake meal, and the meal get absorbed through our GIT, and the different carbohydrates gets break inside our GIT by different kinds of enzymes, and ultimately the simplest form of this sugar is formed glucose, and glucose is absorbed from our GIT and then goes to our liver. So from the liver, glucose enters the systemic circulation, and from the systemic circulation glucose enters into our cell. Now glucose entered into our cell, and glucose is trapped into our cell by an enzyme called hexokinase or glucokinase. This hexokinase enzyme is found in each and every tissues of our body, including skeletal muscle. It is predominant, and glucokinase is found only in specific tissues like liver cells, like pancreatic beta cells, pancreatic isolates of Langerhans beta cells. This glucokinase enzyme is abundant. The function of this enzyme is that this enzyme actually phosphorylate glucose at their 6th position to form glucose 6-phosphate. Another step is that glucose 6-phosphate to fructose 6-phosphate or F6P in short form. This is reversible step, and this step is catalyzed by an enzyme called phosphohexoisomerase. This is a simple isomerization reaction. Glucose is an aldohexose and fructose is an ketohexose. So this is an isomerization reaction. Fructose 6-phosphate is converted into fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate by help of another enzyme called phosphofructokinase. Phosphofructokinase. The name of this enzyme suggests that this enzyme phosphorylate number 1 position. Here ATP is involved. Here also ATP is involved. Where does the phosphate comes? Phosphate comes from the ATP, adenosine triphosphate. At first step ATP is involved, and the third step ATP is involved. Entire glycolysis process is 10-step process. Fast 1 to 5 step. Fast 1 to 5 step is called preparatory phase or energy investment phase, and second 6 to 10 step is called payoff phase or energy gain phase. So here ATP is involved, and ATP converted into ADP, adenosine diphosphate. Fructose 1,6-bisphosphate breaks into glyceraldehyde 3-phosphate and DHAP, dihydroxyacetone phosphate. Dihydroxyacetone phosphate, they are called triose phosphate because they contain 3 carbons. Actual break of this glucose is takes place at the fructose 1,6-bisphosphate level, and this is called triose phosphate. They are interconvertible by triose phosphate isomerase enzyme. Triose phosphate isomerase enzyme. Triose phosphate isomerase enzyme. By that enzyme, they are interconvertible. Glyceraldehyde 3-phosphate enters into another step of the glycolysis. This is also reversible step and form 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate, 1,3-BP, 1,3-bisphosphoglycerate to 3-phosphoglycerate, 3-phosphoglycerate, 3-phosphoglycerate to another step, 2-phosphoglycerate, 2-phosphoglycerate and 2-phosphoglycerate to phosphoenol pyruvate, and all the steps are reversible steps. Phosphoenol pyruvate in short form PEP, phosphoenol pyruvate, and from the phosphoenol pyruvate it is converted into pyruvate, the last product of aerobic glycolysis. The last product of glycolysis or aerobic glycolysis, pyruvate. In the presence of, remember that in the presence of oxygen or in the absence of oxygen, pyruvate production is occurred. But in the absence of oxygen, in case of anaerobic conditions, in case of hypoxic conditions, this pyruvate is no longer able to go into further oxidation process, and it is no longer able to enter into the mitochondria. So this pyruvate is reduced to lactate. But in case of aerobic condition, this pyruvate is further oxidized. It enters into the mitochondria. And undergoes oxidative decarboxylation, and subsequent steps of other metabolic process takes place like Krebs cycle, and if sufficient toxin is available, it is entered into ETC. So this pyruvate is the last product of glycolysis. Here is an enzyme called glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase. This is an oxidoreductase type of enzyme, and here NAD plus participates, and NAD plus is reduced back to NADH. The full form of this NAD plus is called nicotinamide adenine dinucleotide, and formed NADH, reduced version of the nicotinamide adenine dinucleotide. Here another important thing is happened. One inorganic phosphate is added to in that reaction. This is an oxidation reduction type of reaction. 1,3-bisphosphoglycerate to 3-phosphoglycerate is formed by phosphoglycerophosphate kinase or phosphoglycerate kinase enzyme. Phosphoglycerate kinase enzyme. The name of this enzyme suggests that this enzyme transfer one phosphate group, but in this case the phosphate group is transferred from the 1,3-bisphosphoglycerate into ADP and form ATP. This is the first step of substrate level phosphorylation. Phosphorylation takes place at substrate level, so it is called substrate level phosphorylation. In the entire glycolytic pathway, there are two substrate level phosphorylation. So 3-phosphoglycerate to 2-phosphoglycerate. This is an isomer of 2-phosphoglycerate is an isomer of 3-phosphoglycerate. So this reaction is catalyzed by phosphoglycerate mutase. Phosphoglycerate mutase. And 2-phosphoglycerate to PEP is converted by an enzyme called enolase, or simply called enolase A. This is a dehydration reaction. H2 is removed from the substrate, and PEP to pyruvate, another important regulatory enzyme, takes pyruvate kinase participate in the reaction. Here the last substrate level of phosphorylation takes place. So from the PEP phosphate is transferred into the ADP and from ADP converted into ATP. So entire glycolytic process contains two substrate level of phosphorylation. This dihydroxy acetone phosphate re-enters into the glycolytic pathway. This step is called payoff phase. This steps is called payoff phase. Earlier I say that. So this DHAP or DAP is converted into glycerol DH3 phosphate. So two molecule of glycerol DH3 phosphate ultimately enters into the glycolytic pathway. Two molecule of 1,3 bisphosphoglycerate enters, two molecules of 3-phosphoglycerate enters, two molecules of phosphoglycerate enters, and two molecules of pyruvate will be formed. Two molecules of pyruvate will be formed. Pyruvate is a 2-carbon compound. Sorry, pyruvate is a 3-carbon compound, and two molecules of this pyruvate is formed. So NAD plus. So ultimately these two molecules of NAD plus is converted into two molecules of NADH. Two molecules of NADH. So please focus in this section. DHAP re-enters into the glycolytic pathway because this step is interconvertible step. So this NAD plus act as an electron acceptor. Here the entire glycolytic pathway. This is the rate limiting enzyme. Hexokinase and glucokinase. I discussed the difference between hexokinase and glucokinase in different lecture. Phosphohexoisomerase and phosphofructokinase. First step, third step, and last step is the irreversible step and exergonic step, and these steps actually, the enzymes involved in this step actually determines the entire glycolytic pathway, or these steps are regulatory step. The most important regulatory step is this step, the phosphofructokinase or PFK, or simply called PFK1 enzyme. PFK1 enzyme. This enzyme is allosteric enzyme and inducible enzyme. This enzyme is regulated by different factors like insulin, like glucagon, like AMP concentration inside the cell activates this enzyme. AMP concentration inside the cell, ATP concentration and citrate concentration inside the cell, high amount of ATP and citrate concentration inside the cell inhibits this enzyme, so entire reaction will be stopped. And this enzyme, this step is rate limiting step. It means it is the slowest reaction and it determines the rate of the reaction, and this is also committed step. Why it is committed step? Because at this point glucose to glucose 6-phosphate, it is still glucose is not entered into glycolysis. Remember my point, from glucose to glucose 6-phosphate to fructose 6-phosphate, it is glucose still not entered into the glycolytic pathway until and unless this phosphofructokinase works on the glucose molecule. This phosphofructokinase transfer 1-phosphate from ATP to this fructose 6-phosphate and form fructose 1,6-bisphosphate. The ultimate fate of this compound is the glycolytic pathway. So when this phosphofructokinase enzyme works on the substrate, the entire pathway enters into the glycolysis. So this is a committed step, this is rate limiting step, and this enzyme is highly important. Excess AMP concentration increases the activity of the enzyme, ATP and citrate level inhibits the enzyme. Another important factor is that cofactors are also required. Mg2 plus is also required. Here Mg2 plus or Mn2 plus and K plus, some books written K plus, is also important in the last step. Pyruvate kinase is also an important enzyme or inducible enzyme. They are also regulated by insulin glucagon concentration. So this is the entire glycolysis. First, step is called energy investment step. Energy investment step or preparatory phase, simply called preparatory phase. Now calculate the calculate the energetics or energy from one molecule of glucose to one molecule of pyruvate. How many ATPs formed? Glucose to pyruvate production leads to 4 ATP production. This step is 2 times occurs because DHAP enters into the glycolysis by forming glyceraldehyde 3-phosphate. So this is one substrate level phosphorylation from one ATP, another substrate level phosphorylation from another ATP. So 2 ATP. So 2 times enters, it is formed 4 ATP. 4 ATP. 4 ATP molecule. Remember, 2 ATP was first used in first reactions and third reactions in glycolytic pathway. So 4 ATP minus of 2 ATP, ultimately 2 ATP. So one glycolysis gives 2 ATP, but the story is not yet completed. NADH, 2 NADH will be also formed. 1 NADH in modern concept, 1 NADH gives 2.5 ATP. Some books write 3 ATP, but it is not true. In 1 ATH is equivalent to 2.5 ATP. So 2 ATH is equivalent to 5 ATP. 2 NADPH is equivalent to 5 ATP. But this NADH does not instantly give this ATP until and unless they enters into the ETC or electron transport chain, which is not discussed in the which I am not discussing in this lecture. Electron transport chain is another video I discuss this ETC. So when this NADH enters into ETC, it gives 5 ATP. So ultimate ATP production and ETC is occurred when oxygen is present and mitochondria is present. So in case of aerobic condition, glycolysis gives 7 ATP. Glycolysis gives 7 ATP in case of aerobic condition. But if the reaction stops in pyruvate level and mitochondria and oxygen is not available, pyruvate is not further oxidized and ETC is not occurred, this NADH is not reoxidized, and in case of anaerobic condition, glycolysis gives only 2 ATP. But this is highly important because this is the only pathway which works on anaerobic conditions as well as aerobic conditions, and both of the conditions it gives the ATP. So in anaerobic glycolysis ATP production is very less, but in aerobic condition ATP production is high. This is the entire balance sheet of the glycolysis. Now we talk about these two enzymes, hexokinase and glucokinase. Hexokinase enzyme, both enzymes are kinase enzymes, indicates that these enzymes actually transfer the phosphate group from ATP to substrate. But hexokinase enzyme is present in all the tissues and it is predominant in skeletal muscle, where glucokinase enzyme is only found in the liver cells, hepatocytes and pancreatic beta cells. But one of the most important thing is that hexokinase enzyme phosphorylate different kinds of hexoses, glucose, mannose, galactose, etc. And their substrate affinity for glucose is very, very high and their Km value is low. Km is Michaelis constant in Michaelis Menten equation, if you remember that Km. So another thing is that glucokinase enzyme substrate affinity is very low. Glucose affinity is very low in glucokinase enzyme. And glucokinase enzyme is also called hexokinase 4. It is an isoenzyme of the hexokinase. And glucokinase enzyme works well in well-fed state. It requires lots of glucose for working. So when this glucose enters into glycogenesis, glycogen formation, this glucokinase enzyme is required, because glycogen formation takes place when glucose is excess. This glucose 6-phosphate 2 pathway goes on to different pathway like glycogenesis, glycogenolysis, etc. and gluconeogenesis also. Another thing is that when in anaerobic condition pyruvate is formed, and pyruvate in anaerobic or aerobic condition, all condition pyruvate is formed. In all condition pyruvate is formed, and this pyruvate in anaerobic condition forms lactate by an enzyme called LDase enzyme. This step is reversible step. The full form of this enzyme is called lactate dehydrogenase enzyme, and pyruvate to lactate conversion occurs, and in that conversion NADH, which is formed in glycolysis, is reoxidized into NAD plus. And remember, the entire glycolytic pathway, this NAD plus enters into the glycolysis and allows the next glucose molecule to enters into the glycolysis. Please remember the step: glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. This step needs free NAD plus. If all the NAD plus which is available inside the cell is converted into NADH, how glycolysis is continued? Glycolysis is not continued because cells have limited amount of NAD plus. So we need to oxidize this NADH to continue or carry on the entire glycolytic pathway, and this NADH is oxidized by producing pyruvate to lactate by the help of LDH enzymes, and this NAD plus re-enters the reaction and allows the glycolysis. But it is not happening in all times or long time. It is not happening for long time because lactate leads to muscle fatigue or any kind of tissue fatigue. So this is the result of anaerobic glycolysis, and how lactate is formed, why lactate is formed. I explained in the reaction why lactate is formed. So this is the entire glycolysis. If oxygen is available, O2 available, and mitochondria is an abundant, so this pyruvate enters into the mitochondria, and this pyruvate undergoes oxidative decarboxylation to form acetyl coenzyme A, and acetyl CoA enters into the TCA cycle. So this is the entire glycolytic pathway. So thank you for watching this video. Please subscribe my channel. If you like this video, please hit the like button.